hello, namaste. Welcome to Shrav's Biotech for you. Today's topic is competent host. Competent host is the host organism that has the ability of taking up or incorporating foreign genetic material like plasmids or DNA fragments into its genome. This ability is called as competence. Example of competent cells are bacteria, yeast, mammalian cells, etc. This the competent host is essential because DNA is hydrophilic in nature. Hence, it cannot pass through the cell membrane by itself. The bacteria should be competent to accept the DNA. Following are the methods to convert the host cell into a competent host cell. First method is chemical method. Here, the cell is treated with the divalent calcium ion of at specific concentration, which increases the pore size of the cell wall and makes it permeable for the DNA. After that, the cells are incubated with the rDNA on ice and placed briefly at 42 degrees Celsius and 30 degrees Celsius, again put on ice. This treatment of alternate putting on ice and treating it with 42 degrees Celsius and 30 degrees Celsius is called as heat shock treatment. Because of that heat shock, the cell wall becomes permeable to the plasmid or the new DNA and helps in taking up of the rDNA into the cell. Now moving on to the second method, that is physical method. Physical method, also known as microinjection method. In this method, rDNA is directly injected into the nucleus of the host cell. As you can see in the diagram, this is the holding pipette, which along with the small suction force holds the host cell at one place. Then a microinjection consisting of the foreign DNA delivers the foreign DNA directly into the nucleus of the host cell or into a living nucleus of the living cell, oocyte or embryo. Now moving on to the electroporation method. Electroporation method is similar to that of chemical method. Here, the electrocompetent cell is incubated along with the new DNA and subjected to electric shock, that is nothing but high voltage pulse is applied to the suspension of the cell and the DNA. This pulse induces local areas of cell breakdown and pores are formed through which the new DNA enters into the host cell. Now coming to the next method, that is nothing but biolistic gene gun or particle bombardment method. In this method is usually used for the plant transformation, transfection of neurons or transfection of cells into the which are deep in tissue. Helium gas that passes through the gene gun barrel propels it hits the plastic disc, which consists of DNA coated gold particles or tungsten particles. Once it hits the plastic disc, it propels towards the screen. Here, the disc is stopped, but the DNA coated gold particles are further propelled and they hit the target plant cells at a very accelerated speed with a very high pressure. These DNA coated gold particles directly hit the target plant cells and the gene transfer takes place at that moment. With this, I wind up today's session of about competent host. Please like, share and subscribe to my channel.